Hello everyone, welcome back to PhytoPathoVerse. I'm Malini, molecular plant pathologist with the motto to make plant pathology easy. We are here with day 7 of our ASRB NET 2025 revision series. So yesterday we completed the first half of the bacteriology section and today we will continue with the second half of the same section. So you know the deal. This series is all about speed and recall. Listen carefully to each question, glance through the options and try to answer instinctively without overthinking. Let's keep the momentum growing strong. Alright then, without any delay, we will start with day 7. Question 101. Bacteria with one flagellum at each end are known as peritrichous, amphitrichous, lophotrichous or monotrichous. By then you think of the answer, let me draw how this looks. So peritrichous will be the one in which you will see many flagellas surrounding it, okay? Whereas amphitrichous, in this case, we will see two flagellas. In lophotrichous, there will be tuft of flagella on both the sides. And monotrichous, in which there will be only one flagellum. So by the drawing, you might have got the answer till now. So the answer will be amphitrichous, in which we see one flagellum at each end. Okay, but can you tell me one example of each of these types? So I'll tell you for amphitrichous, you will tell me for peritrichous, lophotrichous and monotrichous. Okay. So one such example of bacteria which have amphitrichous type of flagellar arrangement is Pseudomonas species. So it's not mentioned, but it is Pseudomonas species. Okay, I'll tell you one more. Like for example, peritrichous. Peritrichous we have heard a lot of time. What? Ervina, right? Ervina has peritrichous type of flagellar arrangement. One more, Pentuea, or one more, E. coli, right? So for you, I am giving the task of giving me example about the lophotrichous type of flagellar arrangement, bacterial example and monotrichous. Alright. Moving forward, question 102. Bacteria with branches of flagella at both ends of the cell are known as, just now we have discussed, but it's up to you. Go on with the answer. The option remains same. If you are confused, you can check the drawings, which is quite understandable. So, okay. The answer is yes, lophotrichous. Question 103, similar question. Bacteria with several flagella attached all over the cell are known as which one? The option remains same. Several flagella attached all over the cell, all over the body. So this is, yes, option A, peritrichous. Question 104. The endospores forming bacterial genus is Pseudomonas, Bacillus, Ervina or Rhizobium. Answer is Bacillus. Question 105. The symbiont bacterial genus is Pseudomonas, Bacillus, Rhizobium or Ervina. It is Rhizobium, yes. Symbiotic association or relationship. 106. The root nodules forming bacterial genus is Pseudomonas, Bacillus, Nocardia or Rhizobium. The answer is yes, again rhizobium, nitrogen fixing bacteria, right? 107. Bacterial flagella are lacking of protein, antigenic property, 9+2 arrangement or filament. Answer is 9+2 arrangement. So bacterial flagella are consist of what? They consist of 4 main structural units. One is filament, second is hook, third is rod, and the fourth is basal body. Okay. 108. The pili are present in the most of the gram-negative bacteria, positive bacteria, Phytomonas or RLO. RLO is rickettsia-like organisms. Answer is positive bacteria. 109. Lamellae are found in root nodule bacteria, sulfur bacteria, photosynthetic bacteria or denitrifying bacteria. Answer is photosynthetic bacteria. 110. The bacterial 70S ribosomes are present in cytoplasm, mitochondria, plastids or mesosomes. Answer is cytoplasm. 111. The bacterial flagella are 40A in diameter, 60, 12 to 150 or 90 to 110. If I am not wrong, this A is Armstrong, the unit. So, the answer is option C, 120 to 150. 112. Each flagellin molecule is, again, the diameter they are asking 10, 20, 30 or 40. The answer is option D, 40. Okay, but anybody can tell me what is this flagellin molecule? So the flagellin is an incomplete protein containing only 14, 1, 4, only 14 amino acids, and it lacks some essential amino acids, that is histidine, trypsin and more. So okay, that's it. This 14 amino acids is the one that you should remember. Okay, question 113. The basal body of bacterial flagella is anchored in the plasma membrane, cell wall, capsid or capsule. Answer is A, plasma membrane. Moving to question 114. Chemically, capsule and slime layer are consist of protein molecules, fat, polysaccharide, glucose or essential amino acids. Answer is polysaccharide of glucose. 115. The cell wall of the most bacteria is being made up of cellulose, mucopeptide, lipoprotein or chitin. Mucopeptide. Yes, option B. 116. Mucopeptide is a polymer made up of alternating units of NUG and NUM, glucose and galactose, mannose and galactose, and maltose and galactose. Yes. We have heard it. Yes, it's option A, NUG and NUM. 117. In general, gram-positive bacteria contain mucopeptides of 3 to 12%, 20%, 35% or it's 85%. Which one? Answer is option D, 85%. 118. In general, the cell wall of the gram-negative bacteria contain mucopeptide of 3 to 12, 20, 35 and 85. It's same. It's same, the options are same, but see, they have asked here gram positive and in this case gram negative, so it's huge difference. So the answer is option A. In case of gram positive, this mucopeptide will have more amount of mucopeptide, but in case of gram negative it will be less, 3 to 12% only. 119. In electron microphotograph, the cell wall of gram-negative bacteria appears as monogenous layer, bilayer, tripartite or tetralayered. Answer is option C, tripartile. 120. The bacterial cell in which the endospore is formed is known as granules, mesosomes, endospore or sporangium. Answer is sporangium. 121. The stalked bacterial genus is Bacillus, Colobacter, Cercina or Spirillum. The answer is option B. 123. The iron bacterial genus is Vibrio, Clostridium, Cercina or Gallionella. Answer is option D. 124. The non-flagellated bacterial genus is Bacillus, Erwina, Spirillum or Xanthomonas. Answer is option A. 125. The helical or spiral bacterial genus is Nocardia, Christispira, Clodobacter or Vibrio. Answer option B. 126. The helical or spiral bacteria move by contraction of flagella, crista, stalk or pili. Answer option B. Question 127. Most of the rickettsia are obligate, parasites, saprophytes, necrotroph or cell wall lacking. Answer is option A. 128. Rickettsia multiply only within dead cell, virus, living cell or none. Answer living cell. 129. Rickettsia can be cultured in PDA, NA, Richards medium or York culture. Answer is option D. 130. Protoplasts are obtained from positive, negative, rickettsia or phytoplasma. Similarly, I'll tell about 131 because they have asked the same thing. Spheroplasts are obtained from, the option remains same. So, what will happen in case of gram positive and what will happen in case of gram negative? What we will get? Tick-tock. Answer time. So, protoplasts are obtained from gram positive bacteria, whereas spheroplasts are obtained from the opposite of this, gram negative bacteria. Okay then, 132. During multiplication of L-form bacteria by fission or budding, they form large number of small units called protoplast, spheroplast, elementary corpuscle or daughter cell. Answer is option C. 133. Cell multiplication of L-form bacteria take place by binary fission, fission or budding, fragmentation or transverse fission. Answer is option B. Question 134. On culture media, mycoplasmal colonies appear as fried egg appearance, rod-shaped, helical or spiral. Answer option A. Fried egg appearance. Now see, about this question, in most of the cases we study that spiroplasma can be cultured artificially and the culture looks like fried egg appearance. But according to the question and the options given, this is the most appropriate one that we can choose, because spiroplasma is one of the types of mycoplasma like organism, right? That's why. 135. An example of sulfur bacteria is Ferrobacillus, E. coli, Theobacillus or Pneumococcus. Answer is Theobacillus. 136. Extremely resistant asexual bacterial spores are known as exospore, endospore, bactospores or chlamydospores. Answer option B. 137. Cysts are formed in the bacterial genus Bacillus, Clostridium, Azotobacter or Sarcina. Answer C. Azotobacter. 138. Arthrospores forming bacterial genus is Streptomyces, Bacillus, Clostridium or Actinomyces. Answer option D. 139. Linear DNA is found in lambda bacteriophage, phage PM2, phage PM6 or phage this theta 144. The answer is lambda bacteriophage. Question 140. The lipid envelope containing bacteriophage is lambda bacteriophage, PM2, FD or theta 174. Answer option B. 141. In prokaryotes, there is only one chromosome known as plasmid, F factor, ionophore or cofactor. Answer option C. 142. Mutation were recognized in bacteria in 1943 by tetrazolium test, iodine test, conjugation or fluctuation test. Answer option D. 143. Mutation in bacteria was discovered in 1943 by Lederberg, Loria and Delbruck, Kleinberger, Novel or Zinder and Lederberg. The answer is option B. Loria and Delbruck. 144. Bacteria are haploid, diploid, tetraploid or none. Answer A. Haploid. 145. The extra chromosomal elements of bacteria are known as ionophores, chromatids, plasmids or F factor. This we know. Yes, it is plasmids. 146. Bacterial plasmids consist of ssDNA, ssRNA, dsDNA or dsRNA. Answer is double-stranded DNA. 147. The dispensable or autonomous elements of bacteria are nothing but pili, flagella, ionophore or plasmids. Answer plasmids. 148. A mutation which changes two or more different phenotypic properties is known as pleiotropic point mutation, nonsense mutation or frame shift mutation. Answer is option A. 149. Bacterial 6 factor is also known as Cole factor, fertility factor, cryptic plasmid or R factor. Answer is fertility factor. 150. Molecular weight of the most plasmids is in the range of. Please read out the options of this question. The answer is option C. 151. Molecular weight of bacterial R factor is. Answer 1 into 10 to the power 7. 152. Several bacteria liberates proteinaceous toxins known as plasmids, A factor, Bacteriocins or siderophores. So they are Bacteriocins. 153. Bacteriocins are active against other genus closely related strains, L-form bacteria or unrelated strains. The answer is closely related strains, option B. 154. The bacteriocins produced by E. coli are known as R factor, Cole factor, A factor or colicins. Answer is colicins. 155. The term plasmid was coined by Zinder, Lideberg, Dilbruck and Luria or Griffith. Answer is Lideberg in 1952. 156. The bacterial chromosome contains. The options is right on the screen. The answer is option A. 157. Something close to sexual reproduction was discovered in bacteria in 1946 by Lideberg and Tatum in E. coli. The process is known as transformation, transduction, conjugation or recombination. Which one? Answer is conjugation. 158. The transfer of DNA is achieved by conjugation, transformation, transduction or all. The answer here will be all of the above. 159. The number of 6 pili or pili essential for conjugation in bacteria is 20 to 25, 30 to 40, 10 to 20 or 1 to 10. Answer here is option D. 160. Both flagella and pili of bacteria are originated from cell membrane, cell wall, flagellin or none. Answer option A. 161. Bacterial A factor is known as R factor, episome, Cole factor or cryptic plasmid. Answer here is option B. 162. 6 pilis consist of several subunits known as pilin, flagellin, episome or none. Answer option A. 163. Average speed of bacteria is. You can see the options. You know, when they give this range as options, I don't like reading that options. So okay, for this case the answer will be option C. 164. Flagellin is characterized by the presence of. Answer option D. High content of acidic amino acids, low content of aromatic amino acids. Question 165. Epsilon N methyl lysine is found only in flagellin, pyelin, plasmids or sex pili? Answer option A. 66. Bacterial flagellin is a complete protein, incomplete protein, both complete and incomplete or none. We have discussed it on this video. At the first few minutes, we have discussed this, right? So, the flagellin is incomplete protein having how many amino acids? 14, right. Question 167. The basal body of flagella of Colobacter contains 2 rings, 3 rings, 5 rings or 6 rings. Answer is 5 rings. Remember this. 168. Which one statement is correct? M ring is rotor, S ring is stator. Only S and M rings are essential for the function of flagella or none? Answer is none. 169. The energy source of the torque generated between M and S rings is perhaps ATP, total motive force, NADPH or all. The answer here is proton motive force. 170. Peritrichous bacterial species swim in straight line or for some time. Then there is an abrupt stoppage and change in direction. This is known as chemotaxis, phototaxis, trembling or gliding. So this is known as trembling. 171. The toxin produced by Pseudomonas syringae tabaci is known as tap toxin, wildfire toxin, T toxin or all the above. You might be thinking, right? But all these are same. Tap toxin is also known as wildfire toxin, same, and also T toxin. 172. Moco disease of banana is caused by Ralstonia solanacearum, Erwina amylovora, Pseudomonas syringae or Xanthomonas musi. Answer is option A. Yes, Ralstonia solanacearum. 173. Fire blight of apple and pear is caused by Ralstonia solanacearum, Erwina amylovora, Pseudomonas syringae or Xanthomonas musi. The same options, but the answer will be different. Nobody can forget this question because this was the first bacterial disease. So the answer will be Erwina amylovora, fire blight of apple and pear, discovered by T.J. Burrell. 174. Angularly spot of cotton is caused by Xanthomonas campestris pathovar maydis, Xanthomonas axonopotis pathovar kazani, Pseudomonas savastani pathovar malvacearum or Xanthomonas axonopotis pathovar malvacearum. Which one? Tick tock, tick tock. Answer is yes, option D. These are just the causal organisms. Alright. 175. Citrus canker is caused by Citri, Erwina citri, this is Pseudomonas, Xanthomonas axonopotis pathovar citri or Xanthomonas campestris pathovar campestris. Which one? Yes. Xanthomonas axonopotis pathovar citri. Option C. 176. Hollow blight of beans is caused by. Please read out the options, guys. It's right in front of you. I'll just say the answer. The answer is yes, option A, Pseudomonas. Okay. 177. The bacterial genus which causes maximum number of plant diseases, Xanthomonas, Pseudomonas, Erwina or Agrobacterium. Okay, so the answer will be option B, Pseudomonas. Okay. 178. M.K. Patel identified a new species of a bacterial pathogenic bacterium from India in 1948, namely Xanthomonas compestis pathovar physioli, Xanthomonas exonopodis pathovar pallili, Xanthomonas compestis pathovar uppali, or Xanthomonas compestis pathovar glycine. Answer option C. 179. In India, a school of plant bacteriology at the College of Agriculture, Pune was established by whom? M. K. Patel, J. P. Verma, J. N. Chand, or P. N. Patel. Answer option A. M. K. Patel, not P. N. Patel. Okay. 180. Based on symptomology and vector specificity, the pioneering work on MLO disease of plant was led as early as in 1931 by Doi et al., Knockert and Rocks, L. O. Nuckel, or Ishii et al. The answer is option C. Okay. Question 181. Molecular weight of nucleic acid molecules is about 123 into 10 to the power 6 dalton, 0.5 into 10 to the power 9 dalton, 1.5 into 10 to the power 9, or 73 into 10 to the power 6 dalton. The answer is option B. 182. Molecular weight of nucleic acids of bacteria is about, okay, it's dalton. So which dalton? A, B, C, D, which one? Answer is option D, 1.5 into 10 to the power 9 dalton. 183. Size of spiroplasma cells are in the range of 100 to 240 micrometer, 30 to 60, 10 to 20, or 250 to 340. Answer is option A. 184. Spiroplasmas multiply by budding, sexual reproduction, fragmentation, or fission. Answer is option A, B, C, D. D, yes. 185. Bacterial scab disease are caused by species of the genus Conibacteria, Bacillus, Streptomyces, or Agrobacterium. Answer is Streptomyces. 186. The non-root nodule forming nitrogen fixing bacterial genus is Rhizobium, Azospirillum, Frankia, or Rhizobium. Answer is option B, Azospirillum. And this was obviously not Azorhizobium. This would have been the pronunciation Azorhizobium. Okay. 187. Mycoplasma reproduce by budding and binary transverse fission, sexual reproduction, fragmentation, or spores. Answer is option A, budding and binary transverse fission. 188. Mycoplasma are rod-shaped, spherical, gram-positive, or gram-negative. They are gram-negative. 189. Nearly all Fastidious vascular bacteria are positive, negative, spherical, or filamentous. They are negative. Most of them are negative. 190. Fastidious xylem limiting gram-negative bacterial disease are caused by Erwina, Bacillus, Xylella, or Frankia. It is Xylella. 191. The Fastidious vascular bacteria measures. Again, please look at the options of 191. The answer is option B. 192. The gram-positive filamentous root nodule forming bacterial genus is Rhizobium, Azorhizobium, Bradyrhizobium or Frankia? Answer is option D, Frankia. 193. Coconut lethal yellowing is transmitted by Scilla pyricola, Cerulifera tenuis, Mindus crudus, or Dulbulus citricidus. The answer is option C. 194. Coconut lethal yellowing is caused by Phytoplasma viroids, Pyroplasma, or virus. Answer is option A, Phytoplasma. 195. Citrus stubborn disease is transmitted naturally by A, B, C, D. Which one? 195. Option D is the correct answer. 196. Spiroplasma cetri is present within xylem, phloem, root cortex, or apical meristematic tissue. Which one? Answer is option B, phloem sieve tube. 197. Citrus stubborn disease is caused by virus, viroids, Pyroplasma, or Phytoplasma. Answer is option C, Spiroplasma. 198. The constant disease is caused by Spiro, Phyto, Viroid, or Spiroplasma. This was citri, this was Kun. The correct answer is option D. 199. Constant disease is transmitted in nature by which one? Medes, Myzos, Persicki, Bemisia, Tabacea, or Fisgaspi. Answer is Dulbulus Medes, option A. 200. Constant is transmitted in nature by leafhoppers, aphids, mites, or mealybugs. Option A is the correct answer. Coming to 201. The cell division process in rosette forming Colobacter is binary fission, budding, transverse fission, or fragmentation. Answer is option C, transverse fission. 202. The bacterial genus named after a bacteriologist name Bacillus, Xanthomonas, Pseudomonas, or Escherichia. The answer is option Escherichia. Okay. So here is the answer key of this bacteriology section. Let me zoom out. Okay, now you can take the screenshot. Now, in the last video, a mistake was pointed out regarding question number 10. This one, in which you can see your option C has been mentioned as the correct answer, which was lipoprotein, but I have seen a comment in which it was mentioned as pseudomurin. But yes, I really appreciate that. It's great to see such active participation, and in any book, one or two mistakes happens. So I always encourage this type of constructive feedback, and I'm glad you even mentioned the source. And I cross-checked with other reference books, and yes, the cell wall of Archaebacteria contains pseudomurin, not lipoprotein. So in this case, answer will be A, not C. Okay, and okay then, thank you so much for your attention and support. Prepare well for the exam. Until next time, keep learning, keep growing. See you in the next video. See ya.